Have you ever looked at all the blood markers for lipids and thought, what on earth do they all mean? Which one is which, and which are the ones that we actually need to be concerned about? So what happens is we'll ingest some fat. That is then going to be broken down by something called bile, which we release out when that fat reaches our small intestine or the duodenum. And this comes from our gallbladder. This is basically going to just emulsify the fat and get it to a point where these little particles of fat are just so small that they can easily be taken into the cells of our intestines. Once they're absorbed, it then gets repackaged up into something called a chylomicron. Now, in this process, we get some special proteins added to the fat molecule. So as I said, this fat molecule is now referred to as a chylomicron because it's been repackaged up within the intestinal cells. And basically, it's just a phospholipid membrane which have some added proteins to it for recognition and for other purposes, which I'm about to explain. And within it, you're going to have cholesterol you've eaten, you're going to have fatty acids, you're going to have triglycerides, which are just fatty acids attached to a glycerol. And you're also going to have some cholesterol esters as well, which is just basically the storage form of cholesterol. What about the proteins that we add and why? So the proteins that we add are going to be the following: ApoB48 and ApoE, which essentially serve as kind of like a recognition protein, so that when this chylomicron reaches its target tissues, it can be recognized. And the last one is the ApoC2. Now, the ApoC2 is really important, and it's very important that this is functioning properly, because what it's going to do is when this chylomicron reaches its target destination, which is going to be the fat cells, the muscle tissue, the liver, these target tissues are able to recognize this ApoC2, and this ApoC2 is going to be able to stimulate these lipoprotein lipases, which are essentially just special enzymes that are going to start snipping up what's inside the chylomicron. Specifically, what it's going to start snipping up is the triglycerides. Now, as I said, triglycerides are just 3 fatty acids attached to a glycerol. Once it snips those up, they can then be transferred from the chylomicron into the target tissues. So as I said, this is going to be muscle tissue. The muscle tissue might be looking for energy at that particular time, so it can then use those fatty acids for energy production. It can also convert the glycerol into glucose, and again, this can help with energy production. The fat cells might take in these fatty acids and convert them back into triglycerides again, and then we can store them for energy in the future. The liver is then going to take in some of these as well. It can use it for the same sort of things. It can store the fatty acids, it's fat, and it can also repackage some of these products and put them into another molecule. So like the chylomicron, the VLDL is quite a large particle. Why? Because it's filled up with mainly triglycerides and free fatty acids, but it's also going to contain some cholesterol and some cholesterol esters. Now, it also has proteins on its membrane, much like the chylomicron, but instead this time it's actually a ApoB100, which again serves as kind of like a recognition protein. And it's also going to contain the ApoE and the ApoC. Once again, this particle can then go to target tissue like our muscle, our fat tissue, and again our liver, and it can be received by these tissues. It can stimulate with the ApoC2, the lipoprotein lipase, which again is going to snip up what's within that VLDL, namely the triglycerides, and take them into the cells. So as I said, the muscle will use it for energy if it requires it at that time, and the fat tissue is going to take it in and basically store it for the next time we need energy. So as I said, once the VLDL has done its rounds and sent off the fat that it requires to other tissues like the fat and the muscle, it can then come back to the liver. And now the liver, as I said, can use the fat as storage. It can, make some more bile. It can put it in the cell membranes, whatever it needs. What it doesn't need, it then sends back out in another particle. This particle is called an IDL. And the difference between an IDL and a VLDL is basically the size. So an IDL is just slightly smaller than a VLDL. It still does contain the ApoB100 and the ApoE. What it doesn't contain at this stage is the ApoC2. So at this point, because we have lost the ApoC2, we actually gain more of the ApoE. And this is relevant because it makes this particle, the IDL, really attractive to certain tissues, namely the adrenal gland. Now, why is this important? Well, the adrenal gland, as you may have heard of, makes a lot of cholesterol-based hormones. So it makes sense that the adrenal gland loves the IDL because by this point, the liver's repackaged it and it's really lost a lot of the triglycerides that were originally in the chylomicron and the VLDL. And so once we get to this point, majority of what's in this particle, in the IDL, is actually cholesterol and cholesterol esters. Now, as I said, the adrenal gland likes to use this cholesterol in order to build special hormones, and they are aldosterone, which is involved in our blood pressure regulation, cortisol, which, as you know, is involved in our stress response, mainly our long-term stress response, and our dehydroepiandrosterone, which is basically a form of less strong sex hormones that you'd find in both males and females, and they help with creating body hair and things like that, libido. So once the adrenal gland has taken what it needs within the IDL, once again, the particle will then go back to the liver. Now again, the liver can use the components within the IDL, and recycle them, use them in cell membrane production, use them in bile production, can store some of the fat, or it can then repackage back up the remaining cholesterol and cholesterol esters and turn it into the next molecule, which is, wait for it, so the last particle here is the LDL. The LDL, I'm sure you've heard of. It's one of those ones that everybody knows as being the bad cholesterol. Let me explain why this is often the case. So by this point, the liver has repackaged the remaining cholesterol and cholesterol esters into something called an LDL particle. Now, the reason this gets a bad rap is because at this point, it's mainly just cholesterol and cholesterol esters, and the particle itself is rather small compared to the other particles that we were talking about. So the job of this particle, the LDL, is to then go straight to the testes and the ovaries, and it's going to deliver what it has remaining within itself, the cholesterol and the cholesterol esters, and it's going to deliver it to the testes and the ovaries. Why? Because you may not already know this, but our sex hormones, our testosterone, estrogen, progesterone, are all based from cholesterol. So we need that cholesterol to be able to build these hormones. So you don't really want to have no LDL particles because then, of course, you're not really going to be able to give the cholesterol over to these special organs that are trying to make their sex hormones. So in a usual metabolism, what will happen once the gonads have taken what they require cholesterol-wise from this LDL particle, it'll then go back to the liver. And basically the liver will then do what it needs to do with it. It can either store it, as I said, or it can repackage it back out and give it another pass to the organs that require cholesterol. But here's the problem. The particle is so small. If you cannot receive this particle by the liver, this whole process isn't happening properly. A lot of these particles actually can then sort of sit in the bloodstream and just float around. What's wrong with this is that we are also constantly exposed to things that are going to cause oxidative damage. We can be eating these things, we can be eating fats that are actually already partially oxidized, and we can also be exposed to a lot of glucose. Now, this is all normal things, but if you imagine that if you have a lot of these particles floating around in your bloodstream and they cannot be retaken up by the liver itself, they're going to just sit around there and they have more chance of being exposed to the glucose and more chance of being exposed to these free radicals. Now, this is a problem because it actually can oxidize the particles. So essentially, if the particles are in the bloodstream too long, they're exposed to glucose and they're exposed to free radicals, they become oxidized LDL. These oxidized LDL can't be recognized properly at target tissue, and they end up depositing into the walls of our vessels, like our arteries. What happens then? Well, we have these special cells called our macrophages, which are they're basically white blood cells, and they're gonna come to the area and engulf anything that shouldn't be in an area that it's not supposed to be. In this case, it's the LDL particles. They shouldn't be in the artery walls, so the macrophages are gonna engulf them. They then form this thing called a foam cell, which is essentially just heaps of macrophages that have taken up heaps of LDL molecules, and they've just formed this thing that essentially under the microscope looks like a big foam cell. This isn't great because, of course, if you imagine more of these macrophages have engulfed heaps of these foam cells, they're going to cause the artery itself to not be able to be as fluid and move as much as it should do. It becomes quite stiff. This is arteriosclerosis. It can also obstruct the blood flow going through the arteries as well, because of course it's forming this big mass within the artery walls, and this can obviously lead to the blood not being able to reach areas of the body that it should do. But now it's time we talk about the hero of the story and the one particle that I haven't spoken about yet. You might have heard of it. It is our HDL. Our HDL serves so many purposes, and it is extremely important, and hopefully by the end of this you'll realize why the HDL is really the answer to this issue. So what does the HDL do? Well, first of all, once we've made our chylomicron, which you might remember was the first particle we made directly after absorbing the fat, and it did the first pass of the organs and delivered off some triglycerides. But HDL's job is to actually hand over the ApoE and the ApoC2. Again, these little proteins are needed on the membrane of the chylomicron because it helps to recognize the particle at target tissues, and it also helps to stimulate the lipoprotein lipase, which is going to snip up the triglycerides and get them into the target cells. The HDL is also going to transfer these protein molecules to the VLDL, to the IDL, and to the LDL. Again, these serve as recognizing proteins so that the particle can be taken in by the target cells. But what else did the HDL do? So the HDL can also go to the macrophages, which you might remember have basically taken in heaps of the LDL particles and basically formed these foam cells. Now, what the HDL can do is it can actually bind onto these foam cells and basically slurp up all the excess cholesterol that's been stuck inside them. Amazingly, what it can then do is go to the tissues that, again, are requiring that cholesterol, like the adrenal gland, the gonads, and even the fat cells and the muscle tissue, and can actually deposit that cholesterol back into those tissues. This is incredible. It can then go back to the liver, and this whole process can occur again.